Welcome to Catch This. My name is Trudy Ray. In my last video, Catch This episode 76, linked above, I described various traditional vaccine technologies. In today's video, I will highlight 5 more recent advances in the field. These are viral vector vaccines, mRNA vaccines, DNA vaccines, protein vaccines, and dendritic cell vaccines. Don't go away. Genetic vaccines introduce nucleic acids, DNA and/or RNA, into a recipient's cells. Once inside the cells, these genetic instructions are used by the cell's machinery to produce a specific protein. The protein is usually an immunodominant antigen derived from a pathogen, which triggers an immune response specifically targeted at that pathogen. Genetic vaccines include the first 3 types of vaccines I will talk about today. These are viral vector vaccines, messenger RNA or mRNA vaccines, and DNA vaccines. Viruses enter cells by attaching to host cell receptors and releasing their genome into the cell. Viral vector vaccines are based on this principle and consist of a virus, typically an adenovirus, with an inserted gene that encodes an antigen of choice. Several genes native to the vector virus are usually deleted to render it non-infectious and to make space for the inserted antigen gene. Once injected into a person, the vaccine vector will enter cells, and its nucleic acid will serve as a template from which the antigen protein will be produced. The antigen will then prime the immune system to recognize the pathogen from which the antigen originated. When the individual is infected with that pathogen at a later time, the immune system will recognize that pathogen. Several COVID-19 viral vector vaccines encoding the antigenic SARS-CoV-2 spike protein were developed during the pandemic. However, their use in the US was discontinued because they are less effective than mRNA vaccines. The discontinued viral vector vaccines include ones developed by AstraZeneca, previously covered in Catch This episode 4, and vaccines developed by Johnson & Johnson, previously covered in Catch This episode 15. One drawback of viral vector vaccines is that unfortunately many people have pre-existing immunity to the vector viruses. In this case, the antibodies to the vector itself neutralize it before it can deliver its genetic payload, thus reducing vaccine efficacy. Nevertheless, 2 licensed Ebola virus vaccines consisting of viral vectors and encoding the Ebola virus glycoprotein are currently in use, and they are more than 97% effective at preventing Ebola disease. mRNA vaccines gained a lot of attention during the COVID-19 pandemic, but their history is long and complex. These vaccines consist of an mRNA that is encapsulated in a lipid nanoparticle, which protects the mRNA from degradation and ensures proper delivery into cells. The mRNA encodes an antigen, which in the case of the COVID-19 vaccines is the SARS-CoV-2 spike protein. After the vaccine is injected into a recipient, the mRNA enters cells where host proteins translated into the SARS-CoV-2 spike protein, which then promotes an immune response. I made lots of videos about the COVID-19 mRNA vaccines during the pandemic. Check out Catch This episodes 3, 27, and 50. Although the COVID-19 mRNA vaccines are highly effective at preventing severe disease, hospitalization, and death from COVID-19, in contrast to non-mRNA COVID-19 vaccines, their effect does not appear to be very durable, thus driving the need for repeat boosters. The mRNA vaccine platform is also being researched for the development of cancer vaccines, which encode tumor-specific antigens that can train the immune system to recognize and attack cancer cells. This topic was previously discussed in Catch This episode 46, linked above. DNA vaccines consist of a genetically engineered circular DNA molecule containing a gene that encodes a pathogen-derived antigen. Once injected into a recipient, the molecule enters cells and the DNA is transcribed into RNA. The RNA is then translated into the antigen protein, which stimulates the immune system to make protective antibodies. Although several DNA vaccines have been tested for veterinary use, there are currently no FDA-approved DNA vaccines available for use in humans. Nevertheless, this approach offers a number of potential advantages over traditional methods, such as improved vaccine stability, the activation of both B and T cell responses, the lack of an infectious agent, and the relative ease of large-scale manufacture. So we may see at least one of those in the future. Protein vaccines are essentially subunit vaccines that are made from peptides, short antigenic pieces of a protein that can trigger immune responses. You may recall that I talked about subunit vaccines in Catch This, episode 76. The shingles vaccine used in the US and sold under the name Shingrix is an example of a protein vaccine because it consists of a recombinant form of the varicella zoster virus glycoprotein E antigen. Although initial infection with varicella zoster virus causes chickenpox, the virus becomes dormant in nerve cells, where it can potentially reactivate later in life as a painful shingles rash. Shingrix is not only highly effective at preventing shingles, but it may also prevent dementia, as described in Catch This, episode 68. Protein vaccines can also serve as therapeutic agents for cancer treatment, using peptides from the tumor-associated antigens to induce anti-tumor T-cell responses. Protein vaccine candidates for various cancers, including melanoma, lymphoma, and gynecologic cancers, are currently being tested in clinical trials. And lastly, dendritic cell vaccines use the body's own immune system to fight cancer. Dendritic cells are found in tissues that are exposed to the external environment, like the skin and inner lining of the nose, lungs, stomach, and intestines. Once they detect an invading pathogen, dendritic cells process its antigenic material and present it to T cells to initiate an adaptive immune response. To make a dendritic cell vaccine against cancer, one would isolate dendritic cells from the blood sample of a cancer patient. The surfaces of the dendritic cells would then be artificially coated with tumor-associated antigens, and the cells would be injected back into the patient, where they would induce T cells to recognize and attack cancer cells displaying the antigens on the dendritic cell vaccine. The only dendritic cell vaccine licensed for use in humans is Cipla-Cel-T, a therapeutic vaccine used for the treatment of advanced prostate cancer. However, several other dendritic cell-based cancer immunotherapies are in various stages of development for the treatment of other cancers like melanoma and glioblastoma. Although the vaccines covered in today's and the last episode are representative, they do not encompass the entirety of current vaccine technology. Advances in research have yielded numerous novel ideas and platforms, many of which remain experimental. The multitude of effective vaccines available to us is a testament to scientific progress and should not be taken for granted. We live in remarkable times where each day brings the prospect of new discoveries, improved treatments, and better disease prevention. That's it for today. Thanks for listening. Please subscribe. My name is Trudy Ray, and I'll see you next time on Catch This.